before you?
You can just introduce yourself. Yeah.
Go.
Who are you? What's your name?
I am just a scientist, aspiring engineer that has worked on life extension since I can remember. Doesn't want to die. So I'll just leave it at that. A lot of you guys already know me. Somebody asked a question previously about what's actionable about all this genetic data. I would say it's pretty actionable because if I was born like 150 years ago and I didn't know anything about genes, then I probably wouldn't know that I should be working on gene therapy. So just knowing that I have all these faulty genes makes me, the action that I would take on that is to actually develop gene therapy to fix them, not to exercise more, although exercise is fun too. Because, we need to be able to do software updates. Like, we have all this information about the way biology works, and that keeps on growing and growing and growing, but what are you going to do about it? I have this crappy Alzheimer's gene, or, like, I get, like, my eyelashes stuck, and, there's all these things. The motivation module randomly fails to load, like, ApoE, I think, was already mentioned by somebody. If you have the 4-4, then you're pretty much going to get Alzheimer's disease if you don't die of something else first. But if you find out that you have it, you're screwed. There's nothing you can do about it. We need gene therapy. We've known this for a long time.
I've seen those patches. They usually break something else.
Yeah. Yes, there's safety issues as well. So what if we want gene therapy? What is sort of, I'm not sure how familiar you guys are with it, but there's certain sort of things that you can look at in terms of like how good a gene therapy is. And you look at specificity for cell type. You want to edit, if you're developing gene therapy, you want to be able to edit the right cells. You want site specificity, ideally at base resolution. If you want to make a modification, you want to make it precisely. Not like just like say, I'll throw it in this part of the chromosome. You want to throw it at this exact site. Efficiency, you want to be able to modify 100% of your target cells. Size of edit, you don't want limitations on that. You want to be able to make whatever is necessary. Removal of DNA is another thing. You want to be able to remove bad code. You can't fix every problem by just adding code. Although Microsoft tries that. And safety, no off-target DNA modification. So you want to have like high efficiency of your edit, and your edit needs to be precise, but you also need to not have any other things happening accidentally or as a side effect. So you don't want off-target DNA modifications. And lastly, I said live updates. You want to be able to give somebody a gene therapy and they need to continue running. It's like if you try to update your computer, usually you have to reset it, right? We don't want to have to do a reset. So this doesn't really work with biology. You need to keep on functioning during the process. So where are we right now? Most of the sort of clinical trials people have done with gene therapy involve things like retroviruses. Specificity for cells is really poor. Site specificity is effectively random. You're just like throwing it in and it ends up somewhere. Efficiency in terms of what percentage of your target cells, 1% is considered good. Size of edit, 10 kb is about the max, and people generally do much less than that. You can't remove DNA. Safety sucks because most people die of cancer and sepsis from reacting immunologically to the virus. And then live updates, I'd say, like the immunotoxicity kind of makes it not really an effective live update. Things have gotten a little bit better recently. We now have better site specificity using integrases and other things like that. And the safety has gone up in hand with that. So like the better the specificity for a site, the lower the genotoxicity. So those two have both improved. I won't go through the diagram because I don't have time.
Can you tell me what an integrase is?
An integrase is a protein that recognizes a specific sequence and then cuts that specific sequence. Okay. CRISPR is a new version of that, which is much more easy to program. So previously you had to design a new protein for every type of target that you wanted to aim at. Now you can just use it by RNAs. You can just target sites by complementary RNAs, and that makes the editing really much more convenient. And then there's a modification of CRISPR now that uses nicases instead of nucleases. So instead of making a double-strand break, you actually multiplex 4 genome target editing things, and you make 4 nicks onto two strands. And you can swap things out the way that you do molecular biology in the lab. It's very precise. So that's a huge gain, and that also makes things much safer. But we still are like, there's a lot of red up here still. Like this is still not anywhere near the sort of previous sort of requirements that I threw out there. So there's better stuff. And you guys will have to come talk to me about those afterwards. But we can make synthetic viruses now. We don't have to rely on naturally existing ones, and these can be much more powerful. We can do cell therapy. We can just replace cells and modify them outside of the body. Hasn't been done too much in the clinic, but it's coming. And the last slide here, throwing it out there, is really radical. Synthetic endosymbiosis. So people focus on viruses, but there's other things that have entered cells before and carry genetic material. Mitochondria were originally a bacteria. And you could actually potentially make a new synthetic organism that would actually live inside of your cells and have that be the medium for software upgrades. And then here's just some comparisons of those strategies, but I don't really have time to go into it. I tried to cut this slide deck down from an hour to 5 minutes, so it's pretty hard.
You need precise editing.
It's not there yet. The technology isn't there yet.
Cool. All right. Any questions?
Well, what's your thoughts on epigenetics and gene therapy? Are you still beginning to understand that?
Well, epigenetics, like the cell itself, like we have
Like
software.
DNA methylation to turn off genes and stuff.
Oh, silencing.
Yeah.
Yeah, so that's a lot to do with like where you insert it. So if you have precision control of where you insert it, but you can also, if you can do larger edits, you could actually throw in all of the sort of machinery that normally goes with the gene that would actually allow that gene to be controlled on its own, so not reliant on where you put it, but it actually has all of like the enhancers, transcription binding sites for specific histones. And it can have all. You can do a larger edit. You can throw all that machinery in with your software. That would be ideal. But right now, size is limiting.
What do your slides look like in an image, which looked like the logo of like Handle, was it?
Yeah, I'm just friends with them, and they're doing really cool stuff with the delivery of the synthetic viruses.
Do you think, so I see you have their 80% in there, do you think that's scalable for humans across a lot of different things?
We'll know when we try. I mean, that's mice, and it's for a particular cell type, too.
Mm.
Yeah, people have done a lot of things in mice that haven't translated, but I think it's a major step forward, and like, there's a lot of detail there. I'm not sure.
Has anyone else hit high numbers like that with mice for other stuff?
They claim they're the first group that's ever actually changed the genotype of an adult organism, and as far as I know, they're the closest to that. I'm not sure. I would say I think that's a little bit of a stretch, but they're definitely the closest that I'm aware of.
What are your thoughts on telomeres and human longevity?
Cells age in culture medium. If you replicate them and their telomeres get shorter, that's not really relevant in humans because like your somatic tissue doesn't have telomerase active, but it's not dividing, and your stem cells do have telomerase active, and it hasn't telomere length hasn't proven to be very predictable of remaining lifespan. You can knock out telomerase in a mouse, and it takes 7 generations of mice before there's a negative effect.
But to the contrary, when you actually upregulate telomerase in a mouse, they do live longer.
Yeah, but it's probably nothing to do with the length of the ends of the chromosomes. The telomerase actually is involved in some mitochondrial metabolism stuff.
I thought there was a study showing a 4-year lifespan correlation. The longest natural telomeres and the shortest.
I read some stuff that was pretty weak, statistically, and
Well, most studies are like that.
yeah. I don't know.
I read that in a study.
Is there a specific part of the gene that you want to edit it and change it for people in general?
I mean, I have a pretty long list of personal, as soon as it's possible, a long laundry list of things that I'll update. I mean, my ApoE is fine. I guess I could switch from 3.3 to 2.2. That would probably be better, but it's not a big deal. ApoE1 Milano.
Oh, I want
Yeah. Although, interestingly,
all roses.
speaking of statistical significance, it's actually, if you look at the Aspirian trial, it was not statistically significant. It was like moving in the right direction, but the sample size was too small.
This is the family that gets no heart disease, right?
Yeah. But it hasn't been studied enough to say that I mean, I would like to try it, but I don't think it's, unfortunately, it's not, I wouldn't say it's proven or definitely known that that's what's going on.
I'd rather be in the experimental than the control.
Yeah, I mean, it certainly doesn't hurt, so it would be a nice potential good.
All right, thanks, Mark. Okay, were there anyone?
Yeah, who else?
Yeah.
Cool.
I'm on there too.
Yep. Albert.
Yes.
Cool. Do you have a presentation or anything? Nope.